Okay, so now let's talk about what happens to all the other biological molecules. So, biological molecules as fuel sources. So we saw what glucose can do, but what about all the other molecules? So first of all, let's talk about other carbs. Well, other carbs, like other sugars, fructose, galactose, maltose, it's pretty easy for us to understand how most of those can be turned into glucose, right? A lot of them are isomers of glucose. So essentially, we have other carbs. Let's start with polysaccharides. Polysaccharides are basically the starches, the glycogen in animals and things like that. Well, these polysaccharides can be broken down into little chunks. They can be continuously broken down or hydrolyzed and turned into glucose. I'm going to put in other simple sugars. A simple sugar is like fructose, right? Which are isomers mostly of glucose. Polysaccharides are broken down. Usually that's done by hydrolyzation. Oops, that's not going to work. It's too thick. It's a hydrolyzation, or a hydrolysis is the better word. I guess hydrolyzation, I just made that word up. Hydrolysis is usually the process where water is used to break these things down. And once you break apart these long chains of glucose or lactose or whatever form you have, then of course simple sugars, other sugars, can be turned into glucose using various reactions, or they can enter glycolysis at some point. For instance, you'll recall that one of the first stages is that glucose gets turned into fructose, right? And so if you're already fructose, you can just kind of start at that step. You don't need to be turned into fructose. So we can digest fructose quite easily as well, because it just begins to enter the process at that stage. We can phosphorylate it, make fructose 6-phosphate, and away you go. Others can be sort of dealt with by turning them into glucose, and of course there would be isomerases, which are protein catalysts, enzymes that would facilitate the process of turning those into glucose so that they could enter into the cycle. So pretty much all of the sugars and starches and carbohydrates can enter into the process very early on, right at the start of glycolysis, or very near the start. Okay, so I don't know why that keeps going down. It's weird. So that's what happens to other carbs. What about fats? Fats and lipids. Well, remember what fats are. They're glycerol molecules. They seem to be... Okay, so let's go down a bit. Fats and lipids. Well, remember what they are. Remember that fats and lipids are essentially a glycerol molecule. Does that make you think of anything? It's an alcohol, correct? What else does it make you think of? Glycerol. Think about glycolysis. Well, maybe it could be turned into one, I suppose. Some people have. What's one of the stages of glycolysis that sounds like that? Yes. glyceraldehyde 3-phosphate. So this glycerol part can be broken off. So essentially, there's two things that happen. These long chains, I'm just going to draw, well, actually, in humans, triglycerides tend to be the most common form of storage. So let me redraw that as a triglyceride. Okay, and of course these are long chains. So there's two things that can happen through hydrolysis. We can split this molecule up. We can break this up, and we can turn it into glyceraldehyde. Glycerol can easily be turned into glyceraldehyde. And glyceraldehyde can jump into the process at that stage. It's about halfway through, right? It's one of the products that is, it's a 3-carbon. Remember when glucose eventually splits into 2? Glyceraldehyde, 3-phosphate is one of the options. So you can see how that could be sort of shuttled in at that point. These long chain carbons, where most of the energy is in these long chains, so these sections are broken down. So they're digested or broken down by enzymes into little pieces, generally little 2-carbon fragments. 2-carbon fragments. Right? And 2-carbon fragments, where's another 2-carbon fragment in the process? Go through the process of glycolysis, look in your notes, find me the first occurrence of a 2-carbon molecule. I'll give you a hint. You're going to have to look at pyruvate oxidation to find it. Did you find it? It's acetyl-CoA, isn't it? It's the little 2-carbon piece. So when you break these guys up, you can break them up into little 2-carbon pieces that can easily be turned into acetyl-CoA and then can enter into the Krebs cycle. So imagine how many Krebs cycles you could get if you have, 250 little carbon chunks that you've gotten from these long chain molecules. That's why fats are the best energy source. Well, I shouldn't say that. They're the most concentrated energy source. One fat molecule could run the Krebs cycle many, many, many times and produce tons and tons of that NAD and FADH and so on. So that's what happens to fats and lipids, essentially. They get broken down, and they can enter into the pathway at the glyceraldehyde step for the one piece, or into the acetyl-CoA step further down the line. Now, of course, that means that they wouldn't provide any of the stuff before that, right? If they're entering as acetyl-CoA, then they wouldn't have the little bit of ATP produced by the earlier stages of glycolysis, or the NAD that's produced there. But that's well made up for by the fact that they could run the Krebs cycle 35 times for every time one glucose molecule runs it. So there's way more energy in the fats, and that's how they would enter. Then we have proteins. Typically, our body does not digest proteins given the option. Proteins are far more valuable as other things. Question? Okay, actually, that was a good question that was asked. It was the fact that this glycerol molecule in red here gets turned into glyceraldehyde by some fairly straightforward reactions, right? You can add some water to that, and you can make it turn into glyceraldehyde. It's not called glyceraldehyde at that point. It's called glycerol, but it easily becomes glyceraldehyde. So as I was saying, proteins are typically not the choice for digestion for energy because they're so valuable as enzymes, structural components, and hormones, and all kinds of messengers and things that our body typically likes to not use proteins as a source of energy unless we have no other choice. And typically that happens in moments of extreme starvation. If you haven't eaten any food for a very, very long time, your body will exhaust your carbohydrates quite quickly in your bloodstream. Then it will turn to your fats. But once it exhausts those fats, and that could take several months, right? We have enough body fat to actually live for a very long time without any food intake. Water is really the problem. A couple of days without water and you're near death, but you can go months without food because of your fat reserves. But even those will wear down eventually. So in extreme cases where we see cases of prisoners being starved for months on end, we see that their bodies become emancipated. They get all thin and they start to, the muscles themselves tend to disappear. They atrophy and eventually are broken down by the body as a source of last-ditch energy. So a protein, of course, would be broken, first of all, it would be broken into amino acids. Now, if you recall the general formula for an amino acid, it has this amino group on one side, and it has the carboxylic acid group on the other, and then it has a little H, and it has a whole bunch of stuff for the R. So the first thing that happens is a process called deamination, where the amino group is removed. All right? So it's taken away. Typically, I think it would turn into an ammonia or some other. Ammonia is very close to amino, right? Ammonia is NH3. And so ammonia waste would then be handled by your kidneys. It would be converted into urea, as we'll learn later when we talk about kidneys, and excreted as a waste product, more than likely, or possibly be reused to rebuild other things. For instance, you can synthesize some amino acids in your body, so that would not go to waste. So that process is of doing that, getting rid of that, is called deamination. So it's like you're deaminoizing it, right? Deamination. And that can be done through hydrolysis or other simple things. If you look at what's left, though, right? The R group on top, that can be sort of broken apart too. But if you look at this part of the molecule that's left, and you go and look at all the molecules in glycolysis and the Krebs cycle, it looks very much like those molecules. And it wouldn't take much to convert it or change it into one of those molecules, right? They're almost, you can see that it's very, very closely related to those. And so what would happen is this chunk, this chunk that's left over can easily enter into several different stages, actually. It can enter glycolysis if we convert it into some of the pieces of that resemble those molecules in there. It can enter as pyruvate. Pyruvate can be converted into pyruvate. So when I write glycolysis here, I mean it goes into the process. It doesn't mean it changes into glycolysis, because that doesn't make sense. But it can change into pyruvate. It's easy to make that turn into pyruvate. And it can also be turned into acetyl-CoA. Because those pieces are also similar, right? Acetyl-CoA, or any one of the citric acid cycle components. Citric acid cycle components. So proteins can go in all over the place. All right, so what we'll do now is we'll make a little flow chart and just kind of summarize that. Okay, so let's just go down here a bit and make a little chart to summarize all of this interesting stuff. I'm going to use a chart from this textbook. It's quite good. So I'm going to write here in black the stages. So essentially we have glycolysis as the first step, right? And glycolysis is basically where we have I would say maybe half a page you could squeeze it in. So this is the process where we have glucose turning into pyruvate, essentially, right, through many different steps. And then in green up here, I'm going to write carbohydrates, which are all the different starches and sugars, can basically turn into glucose and enter the process of glycolysis at the beginning, or they can turn into intermediates in this process and enter shortly thereafter, like the fructose and so on. So carbohydrates tend to enter the process quite early, right? By either being converted to glucose fairly easily, or fructose or one of the other sugars at the start of that process. After pyruvate, though, we have a conversion to acetyl-CoA, and then, of course, after acetyl-CoA, we have the citric acid cycle. So maybe I'll just put a little cycle here and put Krebs cycle. All right. And then the last stage, which is the electron transport, there's another word for that, which I'm going to use for the first time here. It's called oxidative phosphorylation. I'm going to show you. Phosphorylation. Oxidative phosphorylation. That's, in other words, electron transport in chemiosmosis. So it's describing chemiosmosis. It's describing the process of the electron transport chain and the ATP synthase and all that stuff that we did in that last video.